Hello, I am Dr. Johanna Bendell from the Sarah Cannon Research Institute in Nashville, Tennessee. I am one of the investigators on the CRESTONE study of the anti-HER3 monoclonal antibody Seribantumab in patients with tumors that harbor an NRG1 fusion. NRG1 gene fusions are rare oncogenic drivers that occur in approximately 0.2% of solid tumors. NRG1 gene fusions result in activation of HER3 and can cause tumorigenesis and proliferation. Inhibition of HER3 is a potential mechanism to prevent NRG1 gene fusion-driven tumor growth. The presence of NRG1 fusions is correlated with a poor prognosis, including lower disease-free and overall survival. Importantly, NRG1 fusions are generally exclusive with other known driver mutations. NRG1 fusions appear to be enriched in certain subtypes of cancer, such as pancreatic ductal adenocarcinomas, where incidence up to 6% has been reported. NRG1 fusions appear to be mutually exclusive with KRAS-altering mutations and may be an important treatment-guiding biomarker for patients with KRAS wild-type pancreatic ductal adenocarcinoma. Individual case reports indicate that durable objective responses can be obtained through inhibition of the ERBB family. Seribantumab is a fully humanized IgG2 monoclonal antibody against HER3 or ERBB3. Seribantumab inhibits ligand-dependent activation of HER3 by NRG1, HER3-HER2 dimerization, and downstream signaling through the PI3 kinase AKT and MAP kinase pathways that lead to tumor growth and proliferation. Seribantumab has been evaluated in over 800 patients in prior clinical studies, and its safety profile is well established, with the majority of adverse events being transient and mild to moderate in severity. The CRESTONE study is enrolling up to 75 adult patients whose tumors are NRG1 fusion positive in a tumor-agnostic fashion, and these patients will receive the study drug Seribantumab. CRESTONE is designed with 3 patient cohorts to comprehensively evaluate the activity of Seribantumab in patients who have seen at least one prior therapy. The primary cohort is for patients that have not had previous treatment with a pan-ERBB, HER2, or HER3 inhibitor. Exploratory cohorts 2 and 3 allow patients with either prior treatment with an ERBB inhibitor, patients with tumors that do not have an NRG1 gene fusion that contains an EGF-like domain, or patients that present without sufficient tissue for central confirmatory testing. The administration of Seribantumab is designed to initially halt tumor growth with an induction course, followed by consolidation, and then maintenance treatment at a convenient dosing schedule for patients in order to maintain suppression of tumor growth. The key eligibility criteria for enrollment in CRESTONE is the presence of an NRG1 fusion. Patients with additional driver mutations, for example a KRAS mutation, are excluded. Patients must have at least one measurable extracranial lesion. Local testing of tumors for an NRG1 fusion by CLIA-approved methods is appropriate for initial evaluation. CRESTONE also incorporates centralized RNA-based next-gen sequencing to confirm the presence of an NRG1 gene fusion after enrollment. The primary endpoint of the CRESTONE study is overall response rate per RECIST version 1.1 with independent and central radiologic review. Patients will additionally be followed for determination of duration of response, progression-free survival, safety, overall survival, clinical benefit rate, and exploratory endpoints. The CRESTONE study is currently open and enrolling at sites across the United States. The most current list of activated sites can be found on nrg1fusion.com. The CRESTONE study is being conducted with multiple partners to both efficiently identify patients following diagnostic testing for NRG1 fusions and to facilitate the opening of local clinical study sites in order to ease the burden on patients eligible for the study, particularly given the current complexities around travel due to COVID-19. In summary, NRG1 gene fusions are an actionable driver alteration in many solid tumors, including pancreatic ductal adenocarcinoma. The NRG1 gene fusion appears to be mutually exclusive with other known driver mutations, including KRAS mutations. The inhibition of HER3 represents a rational therapeutic approach for the treatment of patients with tumors harboring an NRG1 gene fusion, supported by both preclinical and early clinical case reports on the activity of ERBB inhibitors. Thank you for your time and interest in the CRESTONE study of the HER3 inhibitor Seribantumab in patients with tumors that harbor an NRG1 gene fusion.